Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Before we dive in, please take a moment to like this video and subscribe to our channel. It truly helps support the Sand Science Experience and allows us to keep bringing you these explorations into fascinating scientific discoveries. Today, we're embarking on, an extraordinary journey into one of life's most universal mysteries: aging. We often think about aging, wrinkles, gray hair, maybe how some organs slow down, but what if we could zoom right in, like really close to the fundamental building blocks, the ones orchestrating pretty much everything in our cells? I'm talking about proteins. And our focus today is this landmark study from Cell. It's by Ding et al., titled Comprehensive Human Proteome Profiles Across a 50-Year Lifespan Reveal Aging Trajectories and Signatures. It's hot off the press from 2025, and it's a really cutting-edge stuff. Imagine getting a shortcut, like a way to be genuinely well-informed about human aging with surprising facts, those aha moments that just click, stuff that might fundamentally change how you think about getting older. We're going to unpack this groundbreaking research. It gives us the first ever comprehensive proteomic map, a blueprint, really, of human organs across a full 50-year aging spectrum.
Yeah, and what's truly groundbreaking here is, we've studied aging from lots of angles, right? Genes, cells. But the complete proteomic landscape, the whole collection of proteins across all sorts of human tissues and spanning decades, that's been a huge blind spot. This study really provides a critical missing piece. It gives us this unprecedented systems-level view of how our bodies age, seen right through the lens of proteins. And proteins are, they're literally the workhorses of life.
Okay, let's do it. Let's open up this proteomic atlas and see what new insights it offers about how we age over time. So maybe start at the beginning. How did the researchers even tackle something this huge? What did this massive study actually involve?
Right, it was an immense undertaking, a real feat of biological data collection. The researchers did this comprehensive analysis, looking at both proteins and, importantly, the tissue structure, the histology. And they did this on 516 samples, which sounds like a lot, and it is.
516 samples. Okay. Where did they get them from?
They were carefully collected from 13 different human tissues. And crucially, these spanned a vast period, from individuals aged 14, so just teenagers, all the way up to 58 years old. That gave them 5 full decades of aging data right there.
13 different tissues. That's incredible coverage. Which parts of the body are we talking about?
Yeah, they covered 7 major bodily systems. So you got cardiovascular tissues, think heart and the aorta, the big artery, digestive organs like the liver, pancreas, intestine, immune system stuff like the spleen and lymph nodes, endocrine glands too, the adrenal gland, white adipose tissue, fat basically, lungs from the respiratory system, skin from the integumentary system, and even muscle. Plus, of course, blood samples, which are always vital for looking at systemic changes, right?
That is just an astonishing amount of data to gather and process. So with this incredibly broad scope, what were the first, maybe high-level patterns they started to see?
Well, first off, the sheer number of proteins they identified was astounding. Over 12,771 distinct proteins. That covers a really significant chunk of all the proteins our genes can code for. And what was immediately obvious was just how dynamic this whole proteome is over time. They saw widespread, consistent changes in protein levels, how abundant they were across almost all these tissues as people got older. If you step back, it's like watching this incredibly complex biological symphony play out over decades. It shows how the very building blocks of our cells are responding to just the passage of time.
Okay, here's where it gets really interesting, I think. It challenges some common ideas. We often think, DNA holds the code, the genes, and that directly dictates everything cells do. Simple cause and effect. But the study points out something called Transcriptome-proteome decoupling. What does that actually mean for how we understand aging?
That's a really profound point. Think of it like this. Your genes are the master blueprints, right? Stored safely. When the cell needs to build something, it makes copies of those blueprints. That's the messenger RNA or mRNA, the transcripts. These mRNA copies go out to the cellular construction sites where the actual buildings, the proteins, get assembled. Now, what this study found is that as we get older, the link between how many mRNA copies are being made and how many proteins are actually produced, it starts to break down. It decouples.
Decouples, so the instructions aren't matching the outcome as closely.
Exactly. The blueprints might still be there, maybe even plentiful. But the actual construction, the protein production, isn't keeping pace, or it's becoming less efficient or regulated differently. And this decoupling was particularly noticeable in some key tissues, like the spleen, muscle, and lymph nodes, important immune and metabolic sites.
So if the blueprints aren't perfectly predicting the final product as we age, that begs the question, right? What is going wrong in this cellular construction process?
Precisely. And the study points pretty strongly towards a general decline in something called proteostasis.
Proteostasis. Okay, break that down.
It's basically the cell's entire quality control system for proteins. It's responsible for keeping all our proteins healthy and functional. It governs everything from how new proteins are made, synthesized, how they fold up into their specific 3D shapes, which is crucial for function, how they get modified after they're made, and critically, how old or damaged proteins are cleared away and recycled. When proteostasis falters, it's like the factory's quality control starts to fail, letting faulty products slip through or not making enough good ones.
And what specific signs did they see? What evidence pointed to this proteostasis system breaking down?
They saw some pretty clear signs. For instance, a decrease in proteins that are essential for just building new proteins. Things like ribosomal proteins that are the actual assembly lines, and enzymes called aminoacyl tRNA synthetases, which bring the amino acid building blocks to the ribosomes.
Okay, so the production machinery itself is slowing down.
Exactly. And beyond just synthesis, they also saw reductions in proteins that are vital for making sure other proteins fold correctly and get to the right place in the cell. These include important chaperonins, proteins like CCT5 and CCT7, which act like guides for folding, and also a key part of the proteasome that's the cell's recycling center, a subunit called PSMB1. All of these were decreasing with age.
And does this breakdown in protein quality control connect to other things we know about aging, like, say, chronic inflammation?
Absolutely. It seems to be directly linked. This decline in proteostasis, this sort of messiness at the protein level. It went hand in hand with seeing an accumulation of certain amyloid proteins, specifically serum amyloid A1 and A2. Now, these are proteins known to build up in tissues when there's chronic inflammation.
Yeah.
And on top of that, they found signs of the immune system being overly active. Things like increased levels of immunoglobulins antibodies like IgL-C and IgHA and complement C4a. Complement C4a is a key player in the immune system's inflammatory response. So this trio, the amyloid, the immunoglobulins, the complement protein they termed it, and amyloid immunoglobulin complement axis. It strongly suggests that this chronic, low-grade inflammation, what people often call inflammaging, is a really crucial feature of aging tissues. And it seems driven, at least in part, by this protein dysregulation.
Wow. Okay, let's shift gears slightly. One of the coolest parts of this research, I thought, was the idea of proteomic age clocks, for different tissues, no less. It sounds like our organs aren't all marching to the same aging beat.
That's exactly right. They developed these highly accurate, tissue-specific proteomic clocks. Imagine, like you said, a clock that doesn't just tell calendar age, but the biological age of your liver or your spleen or your heart based purely on its proteins.
Yeah.
And these clocks could predict the tissue's actual chronological age with really remarkable precision. The correlation coefficients were high, between 0.74 and 0.95.
Right, high. Wow.
Yeah. And it really highlights how differently organs can age. It's very heterogeneous. For example, they pinpointed one protein, TMMP3. It regulates other enzymes involved in tissue remodeling as a key contributor to these clocks in 9 different organs.
9, so maybe a common marker then.
It suggests it might be, yeah, a potential universal marker of tissue aging, or at least very widespread.
Did their analysis show any specific sort of transition points or periods where aging seemed to speed up across the board?
Yes, they did. Looking at the changes over time, their temporal analysis, it revealed a pretty significant aging inflection. A period where many tissues underwent really substantial proteomic remodeling, a major shift. And this inflection point seemed to center around age 50, specifically between 45 and 55 years old.
Around 50, that's interesting, like a biological turning point for our protein machinery.
Seems like it, yeah. A critical window for many tissues.
So if organs age differently and there's this inflection point, were there any particular organs that stood out as maybe early agers? Or maybe acting as hubs that influenced the whole body?
This is a really critical insight from the study, I think. They found that the aorta...
The main artery leaving the heart?
That's the one. Your body's largest artery.
Yeah.
It showed the most pronounced and importantly continuous protein changes across the entire lifespan they studied. It seemed exceptionally sensitive to aging, almost like a canary in the coal mine for the whole system. In fact, the study goes so far as to propose the aorta as what they call a senohub.
A senohub. Okay, that's a new term for me. What does that imply?
It's a powerful concept. It means the aorta isn't just aging significantly itself. It also acts as a primary source, a factory almost, for secreting what they call senoproteins.
Senoproteins, proteins that promote aging.
Exactly. Proteins that promote senescence, cellular aging. And because the aorta is releasing them into the bloodstream, they can travel throughout the body and amplify aging signals everywhere. So the idea here, which is pretty provocative, is that aging in our blood vessels might not just be a result of aging, but potentially a driver that kicks off or accelerates broader systemic aging.
Wow, that flips the script a bit. Vascular aging as a starting point. Can you give us a concrete example, one of these senoproteins and what it actually does?
Sure. A key example they identified is called growth arrest specific 6, or GS for short. They found levels of GS6 were higher in both blood plasma and in the aorta tissue itself as people got older. So then they did experiments. They took human aortic endothelial cells, the cells lining the blood vessel, and treated them with GA6. And sure enough, it triggered clear signs of vascular cell aging, of senescence. They saw more P21 protein, which stopped cells dividing. The cells became worse at forming new blood vessel structures in a dish. And they pumped out more IL-6, which is a known inflammatory cytokine.
Okay, so it affects the cells directly. What about in a whole organism?
Right, they tested that too. They injected GA6 into mice, and the results were pretty striking. The mice showed impaired physical performance, and it accelerated aging not just in their blood vessels, but in multiple other organs too. Liver, spleen, lymph nodes, adrenal glands. It really demonstrated this systemic impact, originating from this one protein associated with aortic aging.
That is pretty significant evidence. One protein having such widespread effects. Were there others like GA6?
Absolutely. Another important one they highlighted is GPNMB. It's a glycoprotein. Similar story. It accumulates in aging plasma and tissues. An experiment showed it could induce aging phenotypes in both the endothelial cells lining vessels and the smooth muscle cells within the vessel walls. And again, in mice, it accelerated both vascular aging and aging in other parts of the body.
So a whole class of these senoproteins, potentially.
Seems like it. They identified several others too proteins with names like CoMP, HGRA1, SLPI, IGFBP7, NGR1, and NODCHE3. All of them seem to contribute to this vascular endothelial senescence, this aging of the blood vessel lining. It really strengthens the case for the aorta acting as this senohub, releasing factors that drive aging systemically.
This whole deep dive really makes you think about the connection to actual diseases, right? The chronic illnesses that often come with age. Did the researchers bridge that gap, link these protein changes to specific diseases?
Yes, they did. And that's obviously a crucial connection to make. They basically cross-referenced the proteins they found changing with age against databases of known disease risk factors. And they found that the levels of 48 different proteins known to be disease risk factors actually increased with age in their dataset.
48? Like what kind of diseases?
Well, it included some big ones. Proteins linked to cardiovascular conditions like APOE and CRP. Both are well-known biomarkers for heart disease risk, but also factors implicated in things like tissue fibrosis, scarring, fatty liver disease, even liver tumors. Proteins like STAT3, TGFBR2, and mTOR were among those linked. So yes, the proteomic changes they observed aren't just abstract markers of aging. They seem directly connected to the onset and progression of many common age-related chronic diseases.
That is powerful. Okay, thinking about the future then, could this lead to, say, non-invasive ways to track how we're aging? Maybe a blood test that tells you the biological age of your organs?
That's exactly the kind of transformative application this research points towards. It's a really exciting prospect. The study actually developed something they called a plasma protein proxy tissue aging clock.
A proxy clock, meaning using blood to estimate tissue age.
Precisely. It's a sophisticated machine learning model. It uses data just from proteins circulating in the blood plasma to predict the biological age of specific tissues, like the liver or the aorta. And remarkably, the accuracy was comparable to the clocks they built using proteins directly from the tissue samples themselves.
Wow, really? That's huge.
It is. It's a crucial step towards developing simple, non-invasive, blood-based biomarkers for organ-specific aging. You can imagine how that could revolutionize clinical assessments and how we manage health as people get older.
Definitely. So pulling it all together, what does this really mean for the future of aging research? And maybe even more importantly, for potential ways to intervene, to actually slow down or counteract some aspects of aging?
Well, this work lays a really strong foundation for a more protein-centric way of looking at aging, understanding it, predicting it, and ultimately maybe addressing it. It opens up some genuinely exciting possibilities for new kinds of interventions, ones targeted directly at proteins.
Like what, for example?
For instance, if we can identify specific aging-associated proteins sitting on the surface of senescent cells, those zombie cells that accumulate with age, it might become possible to develop things like Senolytic vaccines that could train our own immune system to find and clear out those specific aging cells.
Senolytic vaccines.
Wow. Or perhaps even SART-RT cell therapies, like those used in cancer treatment, engineered to target and eliminate cells based on their aging protein signature. Beyond cell targeting, you can think about tackling those senoproteins we talked about, the ones secreted into the blood. Maybe using strategies like PROTACs. These are molecules designed to basically tag specific unwanted proteins for destruction by the cell's own machinery.
Targeted protein degradation.
Exactly. Or maybe developing neutralizing antibodies like we use for viruses or toxins, but designed to specifically block the action of soluble senoproteins like GAS6 or GPNMB. These approaches could potentially block those systemic aging drivers directly at the protein level. It truly illuminates some completely new avenues for improving health and well-being in older adults.
The idea that we could have, Senolytic vaccines or therapies designed to precisely clear out specific aging-related proteins, that's a truly provocative thought, isn't it? Something to really mull over as we think about the future of health. That was an incredibly enlightening discussion, a real deep dive into these comprehensive human proteome profiles. It really clarified the intricate protein-level changes happening as we age, offering insights into maybe why aging feels the way it does, and importantly, what groundbreaking steps might be on the horizon. We really hope this exploration gave you some of those aha moments and helped you feel more informed about the absolute cutting edge of aging research. Once again, please do remember to like this video and subscribe to our channel if you haven't already. Your support is genuinely vital for the Sand Science experience. We'll be back soon with another fascinating dive into the latest in science. Until then, keep asking questions and keep exploring.